And we move to the next speaker, who is Fabio Guglietta. Here you are. So
yes,
just few words. Fabio graduated in Rome, in Tor Vergata. Then he started a European Marie Curie doctorate in 3 different joined university, still in Rome, in Cyprus, and in Aachen. Then he was briefly postdoc at the Helmholtz Institute of Erlangen in Nuremberg. And more recently, since May 2023, he is a junior researcher, RTDA in Italian, at the University of Roma, Tor Vergata. So the title is On the Effect of Membrane Viscosity on Transient Red Blood Cell Dynamics. Please, you can start.
Yeah, thank you. First of all, thank you for inviting me. I think that you can see the screen and also the pointer. I hope so. And
yes.
Okay, great. So yes, today we'll talk about the effect of membrane viscosity on the transient red blood cell dynamics. Because of the heterogeneity of the audience, I will try to make the presentation, let's say, with few technical details, and therefore I will start with an introduction of the complex fluids. That is a topic that is studied, for example, here in Tor Vergata in the Department of Physics. Then we move on to the red blood cells, and we will see why they are interesting. And in particular, we will characterize the loading and relaxation times that are 2 characteristic times of this cell, and also the deformation. We will also talk about the simulation of soft particles. So we will start from drops that are kind of simple system, and then we will move to red blood cells. And finally, a few details in the introduction about the red blood cell membrane. In particular, we will focus on the viscosity. Then there is the section of the results, where I will show you how the deformation, loading, and relaxation times depends on the membrane viscosity. And I will show you also the dependence on the kind of forcing. So depending on how we deform the red blood cell, there is a dependence of these quantities. And finally, a few words about a suspension of viscoelastic capsules. So let's start. What is a complex fluid? It is a mixture of coincidences of 2 phases. For example, the liquid-liquid phases in case of emulsions, or the liquid gas for the foams, or for example, the solid gas for the granular systems, or finally the solid-liquid phases for the suspension. And this is an example, this is a sketch, of course, of the human blood. But indeed, if you look, for example, with a microscope inside a small drop of blood, we can see red blood cells, platelets, white blood cells with different concentration. Most of the concentration is made of red blood cells that are 40%, and this number is also called hematocrit. Then there is the 55% that is just Newtonian fluid called plasma, and the remaining part are platelets and white blood cells. So we immediately see that the red blood cells play the major role in the dynamics of the blood. But what is the complexity of these fluids? Well, I told you that plasma is a Newtonian fluid. Newtonian fluid are fluids whose viscosity does not depend on the shear stress. So on the, let's say, how much we are pushing the fluid, and doesn't depend on the velocity of the fluid. While complex fluids has a nonlinear behavior of the viscosity. In particular, here I reported the shear thinning behavior that is typical of the blood. So it's a behavior where the viscosity decreases at increasing shear stress. Complex fluids have been studied experimentally, but also numerically. And here I reported just 2 examples of simulations performed with the lattice Boltzmann method. On the left, you have an emulsion in convection, thermal convection. And on the right, you have a suspension of red blood cells, indeed, that are flowing in a pipe. Now, why red blood cells are interesting? Here I reported a particular biomedical device that is called ventricular assist device, that is a pump that helps people who suffer from heart failure to pump in the blood in the body. So the blood goes inside this impeller, which rotates at very high velocity, and then it is accelerated through the aorta. These high shear rates in the impeller causes high deformation of the red blood cells, and high deformation can cause the hemolysis, that is the rupture of the red blood cell membrane. So if we want to design these devices to reduce the rate of hemolysis, we need kind of equation that relates the rate of hemolysis here on the left with the stress of the red blood cell. This has been computed experimentally, and there is this exponent that's been fitted with data. But how we do measure this stress? So to do so, we need to perform numerical simulations of this impeller, and then we need something to simulate the red blood cell in order to compute this stress. So we are interested in the deformation of the red blood cell because it is linked with hemolysis, but we are also interested in the characteristic times of the red blood cell, in particular the loading time, that is the time that the red blood cells take to deform. Because if the loading time is much longer than the residence time in the impeller, this means that even though the shear rate is very, very high, the membrane doesn't have enough time to deform, and then there is no hemolysis. How we can simulate such a system? The most simple way is to employ some reduced order models, like the one developed by Maffettone and Minale. In such a model, we assume that the red blood cell is a drop, and whose shape is given by the tensor S, and this tensor is always assumed to be ellipsoidal. So those are strong assumptions in this model. Moreover, we have two external parameters, F1 and F2, that are just two numbers. In particular, F1 here is related to the inverse of the relaxation times. So it's not a kind of ab initio simulation, but you need some external parameters to define this model. And finally, it is one-way coupling. So the fluid enters this equation via the vorticity tensor and the strain tensor, but the drop does not influence the fluid. So this is the simplest model, and we are not happy with this. To make a step further, we can introduce lattice-Boltzmann diffuse interface methods. So now the red blood cell is still a drop, but now the shape is not always ellipsoidal, as you can see from this simulation here. Now it's a two-way coupling because the deformation of the drop impacts on the fluid, because now we are simulating the fluids both inside and outside the membrane via the lattice-Boltzmann method. One downside of this method is that it requires high resolution to avoid this polarized effect at the interface. Moreover, we cannot control the membrane viscoelastic properties. It is just surface tension acting here at the interface, while the red blood cell membrane is much more complicated. So the final solution is the method that we developed also in this publication to study the red blood cell, that is called the immersed boundary-lattice Boltzmann method. This has been widely used in the literature to simulate red blood cells, because now the red blood cell is really a cell, it's no more a drop. Here you can see the 3D triangular mesh used to represent the red blood cell that is made of these Lagrangian points here linked each other to form these small triangles. We use the lattice-Boltzmann method to simulate the fluids inside and outside the membrane. This is a 2D sketch just to make the idea more clear. On these lattice nodes, we simulate the Boltzmann equation, and then this is the interface that the membrane of the red blood cell that can freely move across the lattice. There is again the two-way coupling because the membrane forces are spread to the fluid, and the fluid velocity is coupled with the membrane. And everything is accounted via the immersed boundary method. But most importantly, we can accommodate continuum viscoelastic models on this interface. This immersed boundary lattice Boltzmann method has been developed here in the Department of Vergata in a GPU code. And we used it to simulate different systems, like the drop dynamics in turbulent flows, or the drop dynamics with some interface viscosity. So again, there is an extra complexity at the interface, or to study the wetting dynamics of drop with some viscoelastic properties at the interface. Now, let's go to the red blood cell membrane. So if the red blood cell was like a drop, everything would be easier because we just have a surface tension acting tangentially at the interface, and the shape at rest will be a sphere. For the red blood cell, the situation is much more complicated because the shape at rest is not a sphere, but is this biconcave shape. And moreover, the membrane is a complex structure of proteins linked to each other that create two levels of membranes, the plasma membrane, which acts like a fluid, and it is responsible for the viscosity of the membrane, and the skeleton membrane, which is responsible for the elasticity. In 1973, Scalak introduced this model to represent the strain energy, so the resistance against the strain deformation. If we zoom in on the membrane, we can define the reference patch of the membrane that is measured when the red blood cell is at rest. And then as soon as it deforms, we have a new area. And with these two different lengths, we can define the stretch ratios that are used finally to compute these strain invariants, which enter this equation. The take-home message of this is that the strain energy depends on the geometry, and it is modulated via these two elastic modula that are the elastic shear modulus and elastic dilatational modulus. This is for the strain energy, but this is not the end of the story, because we need to also account for the curvature of the membrane. And to do so, we employ the Elfrich formulation for the bending energy. And again, here there is another bending modulus, another elastic modulus, and this accounts for the curvature of the membrane. Both models have been validated with experiments, and in particular, the elastic coefficients have been measured with high accuracy. Those values are reported here. For the membrane viscosity, the situation is different, it's much more complicated. So to describe the viscous effect, we introduce this Boussinesq-Scriven law. It is a viscous tensor usually used to describe the viscous effect in a fluid. In general, it can be split in two parts. One accounting for the shear membrane viscosity, and they live on the interface, and the other one for the dilatation membrane viscosity, which accounts for the normal, let's say, deformation of the membrane. But since for red blood cells, it's very, very complicated, actually it seems impossible to measure them separately in experiments, they are considered to be equal, and then we just refer to mu m, that is the membrane viscosity. I would like to highlight that this viscous tensor does not depend on the geometry as it was for the elastic part, but it just depends on the velocity gradient. So what experiments usually do to measure the membrane viscosity is to perform some experiments on the red blood cell, and then they measure some quantities, like, for example, the relaxation time with different techniques, and then they use some simplified models to relate the membrane viscosity to the relaxation times. This brings a wide range of values of membrane viscosity, and there is a huge uncertainty in measuring these values. What we propose here is an inverse process. So we fix the value of membrane viscosity, we perform ab initio simulation, and then, depending on the technique that we simulate, we can measure the relaxation time. And by performing a parametric study in μm, as soon as we find the same relaxation time that has been found in experiments, we can fix and find the physiological value of membrane viscosity. So let's go to the results and let's see what we did. What I'm going to show now for the single red blood cell transient dynamics has been published in these three papers, and I worked with these professors here from the University of Roma Vergata, and also with this professor from the University of Aachen. So this is the typical experiment that is called stretching in optical tweezer. So there are two silica bits that are attached at the end of the red blood cell, and they are used to stretch it in this way. And then experimentally, they can compute the relaxation time. We perform the same kind of experiment with numerical simulations, and at each time, we compute the axial and transversal diameters to compute finally the deformation, that is the difference of the two diameters divided by the sum of them. So when the shape is at rest, this quantity is 0, and when it is very, very stretched, this goes to 1. Here you can see the whole process of loading and relaxation. In green, you can see the membrane forces that are sketched here, and in gray, we have the external forces. The deformation as a function of time for different values of membrane viscosity is reported here. The darker the color, the higher the value of membrane viscosity. So first of all, we see that the steady state deformation, called DAV, does not depend on membrane viscosity. What does depend on it is the dynamics. So the higher the value of membrane viscosity, the slower the dynamics, okay? But if we want to measure what are the loading and relaxation times, we need to introduce some fleet functions reported here. They're just exponential. They are called actually stretched exponential because we needed to introduce this delta exponent here. And I'm not going into the details of this delta, but it just accounts for multiple times. But in what I will show you in this presentation, I will just talk about the most important time, which drives the deformation in this plot. So on the left, the loading time, on the right, the relaxation times. We see that qualitatively they behave in the same way. If we increase the membrane viscosity, these times increase. But we see that there is a dependency on the on the strength of the force. The higher the force, the lower both the loading and the relaxation times. Then we compare the deformation as a function of time with the experiments that are these gray points here, and we change the value of membrane viscosity. By doing so, we found that this value here of membrane viscosity matches exactly this curve, and is not reported because otherwise it cannot be clearly seen. And in this way, we could fix exactly the value, the physiological value of membrane viscosity for red blood cells. But now, why there is this difference here for different techniques? Maybe because TR depends on the kind of load mechanism. To investigate this, we performed another kind of experiment. So now it's no more a stretching experiment, but we called it shear simulation, SHS. Again, this is the whole process where we simulate two walls sliding in opposite direction in order to create a shear flow. And we see that the red blood cell deforms, but also rotates. And we compute the deformation exactly as before. So we define the measure axis and the deformation is defined as before. Here, the deformation as a function of time is really different with respect to the previous case. So now the steady state deformation depends on the membrane viscosity, and there are also these oscillations that appear. Again, I don't have time to talk about these oscillations, but I just would like to notice that we need to introduce this cosine here and to introduce also another characteristic times. But then we will talk only about the loading time. So the time that the deformation takes to reach the steady state. If we compare the two simulations, the stretching on the top and the shear on the bottom, we see that the effect of membrane viscosity is not universal. It strongly depends on the kind of mechanical load. We see that for small values of force or small values of shear rate, so in general, for small values of stress on the red blood cell membrane, the intrinsic properties of the membrane arises. It's kind of zero order perturbation theory. And we see that the characteristic times reach the same value, that is 140 milliseconds. But now we want to go beyond this, and we ask ourselves, is there any symmetry between the two processes of loading and relaxation? To do so, we introduce this quantity, T tilde, that is the ratio between the loading and the relaxation times. And we see that this quantity is always smaller than 1. So this means that the loading is always, the relaxation is always faster than loading. And this has energetic motivation because during the loading, there is viscoelastic force and external forces, while the relaxation, there are only viscoelastic forces. But the most important part is that the membrane viscosity does not depend on the, this ratio does not depend on the membrane viscosity for the stretching simulation, while it does depend for the shear simulation. And we want now to focus on these differences because we don't understand now why there is this, there is no symmetry between loading and relaxation for the shear and the stretching. If you look at the two simulations, we see that for the stretching simulation, the membrane does not rotate when it's deformed, and also the force is directly put on the membrane via the silica bits that I showed you before. While for the shear simulation, the membrane does rotate, and the force is not directly put on the membrane, but it is forced by viscous friction with the fluid. So if there is any difference between these two simulations, must be in one of these two aspects. If you look at the velocity gradient in the shear plane, so in 2D, we see that it can be written this way, and this is the geometrical interpretation. And it can be split in two terms, one accounting for the rotation and the other one for the elongation. So if we want to disentangle the two effects and remove the rotation of the membrane, we need to think about the fluid that is kind of elongational fluid which just deform the red blood cell without making rotate. To do so, we perform this 4-roll mill simulation where there are 4 mills that rotate in this way, and they create an elongational flow. So now you see that the membrane is not rotating, it is just deformed, but the force now is not directly put on the membrane, but it's due to the viscous friction with the fluid. The deformation as a function of time looks similar to the stretching simulation, So again, the steady state value of the deformation does not depend anymore on the membrane viscosity, but the dynamics has been slowed down by the membrane viscosity. So qualitatively, loading time, relaxation time behave exactly in the same way. And if we look here at the ratio between the two times, we see that there is again a collapse on the master curve. So for the stretching, for the 4 mill simulation, we had the same behavior as for the stretching simulation. So if there is a difference, is given only by the rotation of the membrane. That this means that when we put energy in the system for the shear simulation, this energy goes both to deformation and rotation, while in the other two simulations there is only deformation. And this is why there is symmetry. Well, there is no dependence on the membrane viscosity for the stretching and the 4 mill, while for the shear simulation we saw a dependency. I would like to stress again that the steady state deformation for the stretching for the 4 mill simulation do not depend on the membrane viscosity. So all data collapse on a master curve for both simulations, while for the shear simulations, we see that there is a dependency not only in the transient, but also in the steady state configuration. Okay, in the last minutes, I will spend a few words about the suspension of viscoelastic capsules, because everything that we saw before was related to the single red blood cell. Now we move to a suspension of viscoelastic spherical capsules, so no more red blood cells. And what I'll show you here has been published in JFM a few months ago with these colleagues of mine. Now, we are not working anymore with a physiological system like the blood. So now we can introduce some dimensionless numbers that are useful to study this kind of system. So we introduce the capillary number, that is the ratio between the viscous forces with respect to the elastic force. And it ranges from 0.1 to 1. Remember that Ks is the shear elasticity in the Scalac model. We also introduced the Boussinesq number, that is the dimensionless membrane viscosity. And it ranges from 0 to 50, where 50 is the physiological value for a red blood cell. We also need to introduce the volume fraction, that is the fraction of the volume occupied by the capsules with respect to the total fluid volume. And it goes from the single capsule, 0.001, to 0.4, that is almost the human hematocrit. And finally, all the quantities that we are interested in must be averaged over all the capsules. And so you will see both the deformation and the loading time as an average over all the capsules. Before showing that, we need to spend a few words about the rheology, because in one of the first slides, I told you that complex fluids show a strange behavior in terms of viscosity. Well, indeed, if we define the relative viscosity, that is the ratio between the suspension viscosity and the fluid viscosity, we see that as a function of the volume fraction, the behavior is not constant. In particular, I also reported the two predictions by Einstein and Batchelor and Green, which hold for hard sphere, so that they don't really apply for our system because we are dealing with soft objects. But still, there is a good, let's say, agreement with the Batchelor-Green, that is a second order approximation in phi, when the Boussinesq number is high. So this means that when Boussinesq increases, the capsule becomes stiffer and behave like a hard sphere. Anyway, this relative viscosity calls for a new definition of the capillary. We cannot use anymore the fluid viscosity to define the capillary number, but we need to introduce the suspension viscosity. So here on the first line, you see the deformation is a function of the effective capillary number, and in the second line, the loading time for different volume fraction. The most important thing is that if we compare the single capsule case with the most dense case simulated, we see that the effect on membrane viscosity here is reduced, because the white points here are the zero membrane viscosity case, while this is the most viscous case. For the single capsule, the effect is strong, while here this effect is reduced. But most interestingly is what happens for the loading time, because here we have a clear difference at increasing Boussinesq, while when the suspension is dense, we see that there is kind of collapse of the points on the master curve. So it means that the Boussinesq 50 simulation behave like the zero membrane viscosity simulation. The effect is completely disappeared. Why this? If we zoom in the suspension, we see that when the volume fraction is very, very high, this is 0.4 case, the particles are very, very close to each other. They even touch each other. And if you remember, the viscous tensor that I'm reporting here depends only on the velocity gradient. And when they are completely touching each other, this velocity gradient goes to 0. So the effect of viscosity reduces. And this can be also appreciated in this plot here, where I am reporting the stress, the viscous stress normalized with the total particle stress. And we see that when the volume fraction increases, so when we move from the squares, the hexagon, so from 0.1 to 0.4, the viscous dissipation reduces. So to conclude, for the single red blood cell, we saw that membrane viscosity must be accounted for because it always affects the transient dynamics in terms of loading and relaxation times. And also, depending on the flow condition, it may also affect the steady state behavior. So even though we are not interested in the transient, but only in the steady state, we need to account for the membrane viscosity. And in general, the two processes of loading and relaxation are not symmetric. When we move to suspension, we see that if we fix the volume fraction, the effect on membrane viscosity is the same as we saw for the single red blood cell. So it's to reduce the deformation and to increase the loading time. But when the volume fraction increases, and in particular when we reached the physiological value of 0.4, that is the human hematocrit, we see that the effect on membrane viscosity and the deformation reduces, and the effect on the loading time almost vanishes. And with this, I conclude my talk, and I thank you for your attention.
Okay, thank you, Fabio. Is there any question? I don't see any.
Yeah, there is a hand from Marco Baiesi.
Ah, Marco, okay, please.
Yeah, okay. So is this set of times, the loading and the relaxation one, depending also on the amount of loading? So if you load more, the times change?
Yeah. We can see clearly here I reported, for example, all the simulations, stretching, shear, and furrow mills. And we see that this symmetry that we are looking, so the ratio between the two times is a function of the strength of the force. In general, it's a function of the stress of the membrane. Yeah.
So is this possible, from these plots, is it possible to identify a linear response regime in which the times are the same? Because I would expect it to be the case.
Yeah, it will be the case when the force is very, very small. So if we report all this data in terms, not of the force, the shear rate, but in terms of the stress, so just to normalize all the data, and we look at very, very small stress, we see all the data that collapse on the master curve.
Okay. Can I ask a second question before leaving?
Yeah, of course.
Okay, I have another question. Do you take into account explicitly the beats in order to compare with the experiments with the optical tweezers? Do you have something representing the beats, or it's...
No, actually the beats are not really represented. What I usually do, okay, I don't have a movie that show it, but what I usually do in the experiments is to select two areas here, and then I apply a force divided by the area just to have the total force equal to the experimental one. And then we need to remove a part of this area as also done in experiment. There is the reference by Suresh in which they explain experimentally how they remove that area.
Because I was wondering whether the effect of the drag, of the viscous drag on the beads is also relevant for comparing with experiments.
I think that, yeah, it might be actually, because, yeah, in experiments this might be, yes. But in any case, we find a good agreement for the steady state where, okay, you don't see viscous effect, but also for the relaxation. And so for the dynamics, we saw a good agreement. So if there is an effect, it's not relevant at this for these forces. Yeah.
Okay.
Is there any other question? Okay, so if not, I think we can stop here. Let's thank the speakers again. Let me also remind you that the next seminar will be on the 13th of June. So good evening, everyone.